We are at 1 o'clock, but we'll probably wait just a little bit longer before we get started. I will remind you, though, that if you could please put in your audio pin if you would like to participate in the conversation. Otherwise, you will be automatically muted, and we can't unmute you. So thank you, and we will get started soon. Okay, so I might go ahead and get started. I see that about 4 attendees have joined. 2 who haven't put in their audio pin when they've called in. I've sent you your audio pin through the control panel. Unfortunately, the interface is such that you come in muted, and the only way that we can unmute you is if you put your audio pin in. So if you can do that, we can unmute you if you have any questions and like to participate in the discussion. So the purpose of this call is to clear up some questions and some confusions that seem to be highlighted in the in-person meeting about what data is available for phenotype data, where is it, and how can one get it. This is something that was very quickly presented on the steering committee meeting yesterday, and will also be discussed on both the genomics and phenotyping workgroup presentations on their next workgroup meetings. And then we will also have another network webinar like this one October 27th. There is absolutely no reason to come to the next webinar if you came to this one. It will be the same one, except maybe different with different questions. As well as this, we'll have this conversation again on the genomics and phenotyping workgroups. The purpose of this is to really help map out some of the questions about what's been done, particularly for the new members of eMERGE. So what we'll talk about is the phenotyping data. So the phenotype data isn't necessarily all held at the coordinating center. The coordinating center isn't so much a data coordinating center, but an administrative coordinating center. But over the last 8 years, there has been a lot of data that's been exchanged and organized and QC'd. So there is some data that is available, some from merge sets or some from specific study data sets that were collected. Also, we will discuss the current available access models that exist in eMERGE, the first being through direct data request, and then also through the existing tools that we have. So as I said, this has been a long grant, and so there's been a lot of efforts over the last 8 years leading up to this point. The first eMERGE grant is oftentimes called Phase 1, and this was the first 4 years of the grant. During the course of that grant, there were 5 primary, 5 secondary, and 2 network phenotypes over the overall GWAS set. This data set ended up being merged and created into a single imputed GWAS set, which included 18,663 participants across the 5 sites from Phase 1. This data was then pulled together in what is called the merged set, which is the dbGaP submission, which included phenotype data, demographic data, particularly case control status across both primary, secondary, and actually network phenotypes, as well as selected covariate data for the secondary phenotypes. So back in eMERGE 1, the primary phenotypes had their own separate dbGaP data submissions that were done by the primary sites that were running those sites, but the secondary phenotypes were not, and so some of the decision was to incorporate some of those covariate data within this big merged data set. And the phenotype data that was collected for that are listed here. And then we moved into Phase 2. This included now 29 more phenotypes. And towards the end of Phase 2, we started discussing again the dbGaP submission across this merge set. When the data was collected and put into a single imputed set, that ended up being about 55,200, actually not about, but 55,029 participants, and this was now across the 9 sites. This discussion was brought to the phenotype work group, and it was decided that the main merged dbGaP submission for the set would include demographics, case control status for eMERGE 1 and 2 phenotypes, which included an update of some of the case and control statuses of the eMERGE 1 phenotypes run across eMERGE 2 data. In addition, BMI and lipids labs data were included in that submission because basically those data points were found to be included in many of the phenotypes run in Phase 2, so we had a lot of data incorporating that data specifically. In addition to the dbGaP submission, over the course of Phase 2, there was lots of study data that was collected and shared across the sites and network. The coordinating center, as part of an administrative task to assist with both the transfer and collection of that data, we, along with the study sites, collected the specific data files submitted for those phenotypes, and that is currently at the coordinating center, although it's not QC'd and isn't merged into NICE study files necessarily. It's the specific site data for specific site phenotypes. But then also, as we moved into the end of eMERGE 2, we also knew and were preparing for collecting clean study data for each of the studies for dbGaP. And part of that discussion was to have some way to do this in an organized manner, because you can imagine 29 dbGaP submissions would be difficult. So we worked out with dbGaP the ability to amend the overall imputed GWAS merged dataset with each of the study data files. And that has been submitted to dbGaP, and that included cleaned study data files for this study specific, and it was amended to the merge set submission. That included you can see here the completed submissions. It didn't include all of the phenotypes in eMERGE 2, mostly because of timing. We had to get the data files in, and some of the studies hadn't completed their work yet, so the idea being that the phenotypes that hadn't been completed would be amended to the dbGaP submission sometime later. That would be cleaned, and those clean study files are in the process of being QC'd at dbGaP and do exist currently at the coordinating center. So in addition to the PHWAS data across the GWAS studies, that occurred in phase 2, we also extended that into looking at the PGX, PGRN sequencing data, and that was a pilot that looked at over 9,000 participants across the 9 sites. And when we were ending the work there, we also decided that that would be integrated into a clean dbGaP file. In eMERGE 2, there were 2 pilots associated with PGX, looking at both lipid labs and ECG data. So there weren't the kind of phenotypes that we had over the GWAS study at that point. So it was decided that the dbGaP data submission would include the lipid labs and ECG data as well as BMI. And that was cleaned and QC'd here at the coordinating center and then submitted to dbGaP. So those data files also exist at the coordinating center. eMERGE tools. So again, in phase 2, we also worked towards making some of this efforts more easier and faster for feasibility. So coming off of that, we created 2 tools to facilitate looking at aggregate counts across the site's data. So that led to the eMERGE record counter and Sphinx. And the data that was incorporated into those were demographics and codes specifically for eMERGE record counter. And that's just a full data dump of ICD-9 codes and CPT codes. And then for PGX, we created a thing called Sphinx, which is the same, very similar. And in that, we also included medications because the PGX study was looking at pharmacogenomics, and we decided that tackling medications and merging of the medications would be beneficial. So that data is available at the coordinating center as it is implemented into the tools. In addition, at the very end of eMERGE 2, it was discussed that we've created these great dbGaP data files with a big status of case and control status across the eMERGE 1 and eMERGE 2 phenotypes. And upon discussion with the phenotyping work group, it was decided to put the case and control status into the eMERGE record counter for criteria, although it was requested to only include the published phenotypes as part of that, that list. So here's just a basic overview of what these phenotype data files look like and are and where they are located. So at the coordinating center, there's the eMERGE 1 merge set, the non-integrated data sets by site and by phenotype for the phase 2 study data, but then the eMERGE 2 combined data set, the imputed GWAS data set that was part of the dbGaP submission, as well as its associated eMERGE 2 phenotype study data files that were amended to that study submission, and then finally the PGX pilot data set. And then, within the tools, there are data sets that include both the demographics, codes, and then medications specifically for the PGX set. And then over here to the right lists the files as they are at dbGaP. And so finally, talking about the how do I access this data. Traditionally, data access has been monitored and governed by the publication policy in eMERGE. So a manuscript concept sheet is submitted and sites express interest in their participation. And then in terms of getting access to the data, if it resides at the coordinating center, we can make the data available to the sites and send that securely. Recent examples of this data, this kind of exchange was, there were several PHEWAS studies on both the PGX and the GWAS sets. And as you can see from the previous slides, the eMERGE Coordinating Center had the ICD codes and the CPT codes, which could be used for that study. So the Coordinating Center sent that to the primary sites, given the permission from the participating sites. And then finally, if this data doesn't actually reside at the phenotype at the Coordinating Center, which you can imagine, there is a lot of data that that wasn't mentioned on these, but in addition, most of these data were, well, actually all of the data was collected at one time. And so in terms of refreshes of the data or getting more updated versions of this, the Coordinating Center wouldn't have that. But the Coordinating Center can facilitate the site-to-site participation request and data exchanges. So yesterday we had this conversation on the steering committee, and it was agreed that some of this data should be made more broadly available. And in terms of that, we've discussed making almost like a data grid of the available case control status across the GWAS studies, and keeping a bookkeeping of what other data may be available. And so from here, I'd kind of like to open it up to questions or comments. Yeah, and I'll remind you that if you had called in and haven't inputted your audio pin, you will be muted. So if you could please put your audio pin in and we can unmute you. Are there any questions? Can everyone hear me? Okay. So this is basically the extent of the presentation, but I welcome questions or any comments or anything like that, and I'll stick around if anyone has anything.
Thank you.
These slides will be made available to everyone.
Thanks, Jacqueline.
Yeah, thank you.
Thank you.
Thank you.